Hi, everyone.
Hello.
Hello.
So this is DCIC group from Miami, Dushita speaking, and I see already a bunch of folks online.
Yeah, so this is Jake. Malvina and Adam are together in my office with me, so they don't have individual icons, but we're here.
Hi.
And
Great.
Vagisha, thank you for calling in. I know you have a
Yes, we
you have a
already-
cutoff in a half hour. So
Right. Yep.
what we may want to do is the agenda all looks fine. We just want to make sure that anything that requires Vagisha's expertise sort of gets covered first, given that she is the primary maintainer and person responsible for the Panorama archive.
Okay, so actually I switched to the slides. I hope you can see it now.
Yeah.
So can we then proceed with the agenda as it is right now? The order of the items, is that correct?
Yeah, I'm just trying to think. As long as we will definitely get to the data processing pipeline and Dockerization, I think that would require Vagisha's input. And so we'll see. We just need to make sure to cover that part before half past, okay?
All right, so let's then start because we're going to have a busy agenda. Let's try to stick to the time proposed here. So let's go then to Yadek to give us a tour about the links Britannics.org site. Is Yadek online?
I don't see Yarek. Hi, this is Mario speaking. I don't see him online. Oh, there you are. Okay, go ahead.
Should, Yadek, can you maybe speak up a little bit louder? Good.
Sounds good.
Should we make you presenter? All right, thank you.
Yep. So, yeah, so I guess the first thing I might suggest is instead of representing this as a circular plot, we just plotting it as a bipartite graph. Like there's clearly an upstream and downstream relationship here. And I think the circle gives it a kind of false equivalency to the sites and the kinases. So that's one just sort of very upper level design consideration. But okay, so now, but what I want to understand next is what is the use case of this graph? How does one then layer quantitative data from the assays onto it? Otherwise, it's just a very kind of relatively static figure. Yeah, I'm gonna reserve judgment on my next comment. But so tell me what the other view is like. I don't think we're going to develop any kind of authoritative proteomics ontology. I think that is outside the scope of what we could do here. I mean, I think there are PSI, the PSI, the Proteomic Standards Initiative, like they're the people who develop authoritative proteomics ontologies. Well, do you want, if you want description, that's text. If you want how to execute the assay, then that I think more fits with the sort of assay specific experiment, acquisition metadata that we've been trying to develop. I think with any of these things, I think what you really want to be driving on is what is the use case? How do you imagine somebody coming to this and asking a question? So I like the idea of sort of coupling that data-centric view to what you call the assay view. I might not call that assay view the assay view sort of from a philosophical standpoint, but the idea that you could have something interactive where you can click on a profile or select a group of profiles, and then if you had a bipartite graph that showed all the analytes in the assay, and then either they're known or predicted upstream kinases, you could kind of represent that biology. That would be very cool. Yeah. Yeah, you need to collapse that. That's too confusing. Like all of the hist H3, A, B, C, D, hist 1, H3, A, B, C, D, blah, blah, blah. Like that needs to be collapsed to one node. Only in sequence regions where you can differentiate more than one proteoform should you bother to replicate that. Yeah. Yeah. I mean, I would just explicitly link these views already from the beginning. I don't think I would necessarily divide them. So I have to think. I have to think about it. Something doesn't readily come into mind, but it doesn't scream assay to me. That's the information that you're showing doesn't necessarily suggest assay to me. Okay, look. I want to make sure that we talk about the Dockerization thing before Vagisha has to leave, and she needs to leave in like 8 minutes. So maybe we can come back to this, but I think that is key. Tell us first what you're trying to achieve by Dockerization of the pipeline.
Yeah, so Jake, I don't know if you heard, we discussed this yesterday during DWG meeting. I showed a few slides about where this is going and why is it being done. And I can show you this quickly again if you didn't hear about it.
So I wasn't on the call. Adam, who's with me, is on the call. So he's seen it and could fill me in. And again, anything that requires Vagisha, I want to make sure that she's here for it. But it sounds to me basically like you want to have Dockerized instances of running any sort of processing protocol.
That's correct. And again, you know, I made this, tried to make this clear. It's not we wanted. It's so we were nudged in this direction in general by Ajay and the crowd. But now it's being made as a requirement, right? And it's coming from NIH, and they want to have every processing pipeline Dockerized.
Okay, so what particular parts of our data processing pipeline do need to get put into a Dockerized instance?
So I guess the one that we dealt with was processing of that you know the Bethru's went and tested. You remember that interaction, right? So maybe
right. Now they're validated.
Right, exactly, right. So it's officially now validated, and basically now needs to be Dockerized. And I mentioned this yesterday. There are 2 ways to do it in principle. You guys can do it, or we can do it. That's the bottom line. And there are pros and cons for both of these, right? If you do it, you have complete control of what's being done. It's a smoother integration. If you change something, right, you can immediately trigger recreation of the Docker and so on. If we do it, you know, we kind of have protocol already how we organize these. It may be relatively straightforward for us to actually do it, and it's still again, I can show you how it works with the one that we already created for BIMA pipeline. But if Adam saw it, maybe he can tell you how this was done.
Yeah, I think, you know, you'll post the slides to the DWG website, and Adam can fill me in. So I don't think that's a good use of time. But the idea is that people would be able to download a Docker instance and then download, you know, data, you know, raw-ish data, unprocessed data, and just redo it themselves? Or is the idea that they'd be able to download this Dockerized version and then manipulate the parameters within the Docker container to change outcomes?
Well, I mean, so I guess the Docker is supposed to facilitate the original pipeline processing. What people do with the Docker, that's up to them. They presumably, it provides them with a complete system with all software and everything there, and they can do what they like to do. If they, I think, I didn't really think about this particular question, but I think once you have the Docker container and completely transparently created, right? Everything is clear, what's in the container, how to interact with this container, and so on. They can also modify things for their own purposes, right? And play with, say.
Okay, so there's three important things to know about where we're going with our processing pipeline. One is that we are deprecating the R version of our pipeline, and we're going to go Python only going forward. What this means is that we're actually, Vigisha has been working with another person in our group, Lev, to work on a more of a handshaking system between Panorama and the servers here at Broad. So right now everything is done in the Panorama server. The pipeline runs in Panorama for what you see today. But in the future, the Broad server will pull the Panorama server to see if there's new data, and if it finds new data, it will slurp it down, process it, and then upload all the output files. And so there's going to be a little bit of a reorganization of the Panorama views there. And then the third thing is that we are, and I'm sure Baruz and Yarekel will chuckle at this one, is that whole little JSON thing with the processing parameters. We're in the middle of reimagining how that's going to work. We sort of think that while I like it, Lev, from a software engineering perspective, has pointed out its numerous deficiencies, and so that's probably going to change. And with that, there is on the roadmap a future version of the GCT file format that we use called, it'll just be a rev number GCT 1.4 that will contain not only sample and analyte information, but probe, or not sample and analyte information, but also sort of document or experiment, whole experiment level information. And we envision that we will include some of the parameters for the processing pipeline in this sort of document level representation of the data. So there are a couple of things juggling in the air with respect to our processing pipeline. And so what's your opinion on the right time and place to Dockerize something? I would just as soon have you Dockerize it. I would say that you can Dockerize our current pipeline as it is, and when we're ready for a major rev, we would let you know, and you can Dockerize that one.
I think that sounds reasonable. And then this way we stay on the timeline, right? We keep pipeline officially validated and all these kind of things without opening this kind of worms again, and just Dockerize what you have. So I think that sounds very reasonable.
Okay. So at this point, there's no action item required from us on Dockerization. You'll take what you have and have validated and Dockerize it.
That is correct, right? Yeah.
Great. Well, thank you, Vigisha, for attending. If you need to take off, feel free.
Yeah, I do. And actually, it would be great if I could get a copy of those slides as well from
The
Mario.
DWG
Yes.
slides. Good.
Yes, that would be great.
Yeah,
Okay.
we'll post it on DWG. So
Okay, great. All right, thank you so much. Bye.
So should we move to the next agenda item?
Sure.
here. Let me go. I think the CRISPR data set. So basically, what is the plan with that and the timeline?
Yep. We are planning, we actually reviewed the last of our CRISPR data this morning. We're planning to have all CRISPR data sets that we're going to do for the time being released by the end of this month. And I think at that point, it would be appropriate to package. Yep. Yeah. No, that is true. That is true actually, and it is a superset, right? So it includes all of the ones that are in the earlier thing, but we upgraded the assay to look at more marks, and so going forward, we have more marks. Blank room. Well, I think it is our opinion that the CRISPR data should be packaged as their own thing, separate from the chemical data, and therefore there's really no problem, right? Because all the CRISPR stuff will have these H4 probes in it, whereas all the chemical data will not have it. You could still use any downstream tools to compare one or the other as long as they only worked on common analytes. The issue will come to a head when our next round of chemical perturbations, which is sort of in progress now, gets released, and they will have now these additional probes. And so whether you jump them together with the old chemical perturbations that are the existing chemical perturbations, that's when it will sort of come to a head. Now I will say that if you look in the provenance codes of the CRISPR data, you will see that the first token is also different. Whereas in the chemical perturbation data, the first token is GR1. Now the first token in the CRISPR data is GR2, indicating that we have revved the assay by one full revision. So that's how to do a sort of assay compatibility check, is by looking at the first token in the provenance code. Just GCP. Yes. Yeah, but we've got a little time on that front.
All right, let's move on. The next agenda item, it's the always fun metadata. So small molecules, we have discussed this in the previous meeting, some of the issues, and in the meantime, we have exchanged like a lot of emails and documents. However, we still have some open questions like how to proceed. So let me just go briefly. For you guys who didn't see that document with like all the details about every single challenge that we are facing here. So the first one is the batch sample ID. So far we have been generating dummy SM Center batch ID in the form of the PCCSE and the drug name provided. And our proposal to move forward is basically to utilize the ADAMS registration system for both the small molecule center canonical ID and small molecule center batch ID. Do we have any comments on this, or we should consider this agreed upon?
So just so we're all on the same page, this is what Malvina is like our internal ID. It's like a number in the range of like 1 to 800. And so this would just be replacing the BRD IDs. We would not, farther along the line, we would not actually remove the BRD IDs from Skyline files. This would simply happen like behind the scenes. So this would now, our internal ID would match to an LSM ID, which would totally eliminate the BRD mismatching problem we've been having. But we would not have to go back into any of our prior Skyline files and re-annotate. All we would have to do is just send the DCIC these internal IDs. They would then attach those to the LSM IDs, and then we would be good to go. And we would no longer have to
So you'll create a new sample metadata field in our data that will be appended to all of the metadata. It will not replace the metadata field called PERT ID though, right?
Correct. It actually, we won't put it in. Yeah.
Yeah.
As I understand it, Dushitsa.
Is that correct? You will append a new sample metadata field to all of our data that just captures our internal batch identifier.
Or it would be linked to the LSM ID. I
Okay.
believe we've discussed that this internal ID was going to be linked to the LSM ID, correct?
Right. The both batch ID and the canonical ID are linked to the LSM ID after the standardization, right? So basically, we're following the links standards, and these are exactly how they are named, like this small molecule center canonical ID and small molecule center batch ID, right? So for that, I can bring up the proposal quickly that has like a few examples here. So here it is. So basically, we will use this system now. Here it is. Right? So we will have a name as provided, and then we will have this canonical ID, which uses the Adams system, how you guys track it internally. So 50 will correspond to the canonical level information, which is the linostat here. And then the 50-1 would correspond to the specific sample of the linostat. So that will be, that's how we will have it in the metadata.
Yeah, this is all totally fine as long as it's clear that you guys are appending these fields into stuff that you harvest from Panorama.
They're going into the metadata files. So we are trying to leave the data files unchanged. So basically all additional information, not just that, but like you know, for example, CABI IDs and other stuff that we are adding to the metadata specifications will be part of the metadata files. So
this actually, Dushitsa, I think Jake brings up a good point. Is like, is this internal ID field, the 50-1, is that ever added into the metadata as sitting on the links data portal?
it will be. It
And then the
will
question is,
be batch ID.
it will be batch ID, and is that not sure. Will that be? I don't know all the internal workings of how that exactly happens, but would you need to, let's say we have a Skyline file that's on our Panorama server we released like 2 years ago. Would we then have to go back and change our metadata fields in that Skyline file, or would you be able to just do that by linking to the LSM ID when you harvest the data?
Actually, I have that on the slides. That's one of the challenges too. So maybe we should discuss that because we have a few proposals about that. So ideally, let me go back to the slide. So let's suppose that this is how we should proceed.
So, can I make a comment on this? I mean, I think this may be a way to do it, but I would recommend to you guys, if you want to use this moving forward and beyond just looking at, completely like, very well characterized drugs that have a very distinct name, right? And where it's pretty clear what it is. this type of registration and assigning IDs will not work, right? Because you will get the same problem that you're going to end up having like two slightly different structures. They're going to have the same name, right? And then the whole idea of having a canonical ID is basically that defeats the entire registration ID, right? So if it will probably work for the most common drugs, you know, where they're very well characterized and it's pretty easy to find what they actually are. But if you use it for anything else, you know, some new compound somebody made, you know, there may not be a clear name, or, you know, in many cases they're like, you know, people use the same name for EZ isomers and that, and it's not always, you know, specifically annotated. There will be problems, right? Because you will end up having the same ID, and then if you mapped it to some other source that uses a structure-based ID, then you may just end up having two different of those external IDs corresponding to your internal canonical ID, and that kind of defeats the purpose, right?
So you're talking about broad IDs, actually. Then.
Well, that, or, you know, like if you create an ID based on a name, it's typically not a very good idea, right? I mean, you need to have, you need to do it based on identifying a particular, you know, chemically unique entity, right?
Yeah, so I think, you know, we agreed on what the move would be going forward for any new compounds, which is we're just going to send you the packing slip or with the lot number and whatever identifying information, and let you assign for any new compounds.
You can do that. If you're happy to do that, it will probably be ultimately better for all the groups, right? And we will do that, and we will send you back the structure, of course, and the LSM ID. My comment was just meant to, you know, if you're building infrastructure or resource that you plan to use, like, you know, many years to come, right? It's probably worthwhile to think it through a little bit more. But again, I mean, it was just a comment, right? It will work for links, you know, the way Tushitsa will propose, we will just proceed. But if you want to use the same one, then, you know, for other problems, for other projects where some other folks may give you compounds and you want to register them, and that, you know, this could be, I could see that may not work for all cases.
Okay. I think our primary focus with these particular identifiers is to make sure it works for links, which is why we want to put it into your hands. I do take your point. I do take your point that this is not a global solution. You know, we thought we had that solution here internally at the Broad with the BRD IDs, but we see now that we have multiple BRDs that map onto the same compound, and so that's our internal challenge to solve.
Sounds good. So yeah, let's do that. You know, I mean, again, I mean, so we ask you just one other quick question. So can you like estimate the number of compounds? It will be around 100 or something like that, right? To curate from those vendors, you won't end up screening thousands of compounds, right? Or can you estimate that?
Yeah, it would be in the hundreds, not the thousands.
Okay.
And in fact, you'll be so happy because our next batch of compounds are all ones that the Detox S Center has already profiled, and they already have LSM IDs.
Oh, good. They're already
we actually got the vials of compounds from them. So they're really the same compound.
Yeah. Perfect. We actually manually curated them from them too, because there were many of them were not right. So,
so we have the LSM IDs for all those, and we're actually going to probably send you, hey, we've got these LSM IDs. Send us back all of the information that you have curated from them, so that we can, as much as possible, incorporate the right thing.
Okay.
Okay. But now, in terms of the implementation, right, to put in the sort of dash notation on the batch IDs, right? So is the proposal for us to send you a big table that basically has our sample identifier, which is this kind of unique, you know, nonsensical looking string, and then map onto it the internal identifier and the internal batch ID, and then you will, behind the scenes, basically merge or join this into the data that you scrape from Panorama.
Well, this is actually where we say here that, okay, so we already actually have this issue that the data is not synced, right? So ideally, after we finalize all these IDs, like have this internal PCCSE ID for both batch and canonical information, ideally the file should be updated with this finalized IDs.
So I think what Tushita is saying, that it would be better if you have very clear provenance, right? And we wanted to follow like a process in which we essentially do not change the files that are released, but just add additional ones, right? And then add the mapping information, and then from those the derived files can be produced. So
yeah, I think but I think based on what I proposed, you could do that. So the thing that's already sort of immutable in the file is the sample ID, right? So if we give you a table of mapping of sample ID to a new canonical batch ID for each compound, you can just join that in. Like we could generate that table, right, Adam?
Yeah.
Like every, it would be basically for all the existing data so far, it would be about a table of 5,000 sample identifiers with their canonical or center ID and center batch ID, right? We could do that.
So basically remapping to this new type of the batch ID, right?
Yes, exactly.
Yep. We just need to produce that table. And so then we don't change our original data files. There's an immutable token in there that represents the sample, and you just join on that token.
But I still, I'm sorry. So are we gonna replace the sample IDs, the sample batch IDs? Yeah?
Oh, we can't do that, unfortunately.
Okay.
So basically what we're saying, for the files that are already in Panorama, the data files are gonna stay the same, not updated, but you will provide this additional mapping file that's gonna map the unique IDs from the current data files to this newly proposed Atom system based batch ID, correct?
Correct.
Okay.
And then you want, I understand it's hard to change them in Panorama, I get it. But when we put them into the data portal, we want to replace the sample IDs?
Well, we can
We're
never
gonna add another one.
It wouldn't replace them because then they don't map back to Panorama. Right? It's not a good idea.
In the mapping file.
We can put it in the mapping, or maybe we could even think of if there's a way to put two batch IDs if it's really the same batch, right? The same exact sample.
Well, I guess now so what we currently have registered with you is what we call a BRD ID. So it starts with BRD dash, then it's like K or A or Q. Those would now no longer be registered through links.
Yeah.
Those would only be for our specific internal processing. And then now the only thing that would be registered would be the LSM IDs. So essentially, from your perspective, now these BRD IDs would just be something extra.
No, I know, but those are canonical IDs. I just wanted to clarify the sample IDs first. The sample batch IDs. The broad IDs, they were canonical IDs, right? Your internal canonical IDs.
I see.
Let's just first clarify. Really, for me, I'm sorry. So that what are we gonna do exactly with the sample batch IDs? So right now we have the PCCSE a drug name version, right? And now those will be replaced in the release data files except in Panorama, or we want to add the new ID, the new system to this, right? In some way.
So in our current metadata that's embedded in the Skyline file, the only thing that we have is something that's called PERT ID, and that's typically a BRD number. You guys, I think, have been mapping that onto one of the official links things. We also have LSM ID, of course. We have LSM ID. For the limited number of conditions where the LSM ID needs to change, that part we can try to go back and do.
Okay.
But we're not going to go in and now create and append a new thing, which we would call per center ID and per center canonical batch ID, right? Like you're already mapping from something that we have named one thing into the official links nomenclature, because of course we had these field IDs developed before the links official nomenclature was really locked down. So I think what Adam is suggesting is no longer take our PERT ID and map it. Well, all you're going to do is you are going to map off of the sample ID, which is this sort of unique identifier. We have it in here somewhere.
Okay.
And just map that into the canonical center ID and canonical center batch ID. Does that make sense?
Okay. Yeah.
Where is ID? Oh, there's ID. Okay, yeah. It's just they don't know about that. What ID?
You know, maybe for the batch ID, maybe you even want to include the code for your vendor. You know,
We do. In these internal IDs, yes.
so that then there's really no ambiguity possible. You know?
Yeah.
Because it doesn't matter if they're a little bit longer, if they have a few more codes, you know, it's fine.
Right.
But yeah.
But yeah, that makes sense.
Yeah, just decompose the sample ID.
So we will then repackage the already released data. We'll add this mapping file with the new internal PCCSE IDs, and that should take care of the mapping. And then we're ignoring road IDs, correct?
Wait, hold on. I think we're both having sort of internal conversation here. So Adam, just state again.
So I think maybe Jake, you and I think maybe the three of us aren't all on the same page here. So this. So Jake is talking about something that we call a sample ID, which is a unique identifier for every single injection that we run. So every single sample in a set of 96 has a unique sample identifier. So there are 96 possible entries for that in one batch, one plate that we call it. The LSMs are, because we do our analyses in triplicate, the LSMs are then, there's only 32 unique LSMs in a plate because we do triplicates within a batch of plates. So we add as an internal metadata field in our Skyline documents this sample ID. We simply call it ID, and it's a human irreadable string that says exactly what cell line, exactly what day it was generated, and exactly what well it was generated from of a 96 well plate. And so Jake was talking about using this field to map onto the LSM ID, whereas we had been talking, Dushitsa and I, about using the internal identifier, which is this much smaller, like 50-1 token. Yeah.
Yeah, no, I'm saying take from the ID and provide a list of 50-1s.
Yeah.
Okay.
Map, yeah, I think we are on the same page. Every sample will provide the compound enumerator, the center compound number. It'll just be a merge. It'll be a simple database join if you want to append these new things. And we will double check that the LSMs are mapped correctly through that process.
Okay.
Yeah. Yeah, do that and keep the internal hierarchy, of course, right? So obviously.
Yeah. But the one thing that we know is what drug we put on which sample, and so that's the basis for conclusion. Okay. Look, it's 5 up. I do have a super hard stop because I have a really key meeting coming up at 1. Let's quickly touch on the experimental metadata. Okay. So can you just pull the assay specific metadata very quickly? And I just want to show you what I've done, but I really have to go at 1. Okay, right? So the idea here is if you even start 1 tab to the left of this, just the general proteomics tab, okay, we try to find what are just the least common denominator of things for proteomics experiments, and it really comes down to this very limited number of fields. And then the thought was to have then assay-specific sort of further information. So now you can move over to the right, proteomics targeted. Okay, so this is, I think, the minimal set of fields needed for a mass spectrometrist to understand what was done to instruct the mass spectrometer to acquire data for a targeted assay. I've just basically given a proposed name, definition, type, if there's a controlled vocabulary, and then what it would look like for the GCP assay. Right, and then there's a key feature of this, what's the last field is called a target table, and so that would be the next tab to the right. And again, for a mass spectrometrist to understand exactly what the mass spectrometer is doing, this would be the preferred way of specifying that. The thing that I wanted to have in here was the PLN for the actual analyte, and right now I don't think we're quite there yet. But that would be my desired outcome. Is this what's column B, analyte protein line notation should contain that. Okay, fine, and we'll just want to make sure that it's representing the right level of information. It's not overly specific, but it connotes the thing. So then continuing on the proteomic swath DIA, right? This is a very similar set of fields. There are just some small differences. Again, these are designed for a mass spectrometrist to be able to understand the data acquisition strategy that was done. And instead of having a target table, you have a window table that you would use in swath. And for this example, we have it completely filled out for the P100 assay. So I think the next step here is to release these for comment to the rest of the folks. I already told Mike McCoss to be on the lookout for this, so the rest of his lab can take a look for it, but to engage Jenny on the swath DIA to make sure there's not some little bits of it they want to left out. But I mean, as far as I'm concerned, they're at a level now for next level review.
So should we just share it with the proteomics working group?
I think no. You want to be a little bit more targeted than that. For example, you want Jenny and her group to review the proteomics swath DIA specification. You might want Peter Sorger's group to review the proteomics targeted specification, because some of the work that they're doing is highly targeted. And then it's on the other groups to define the discovery proteomics or whatever other flavor of proteomics that they want to define. If they could follow this general template, it would be nice.
Okay, maybe
Great,
the data working group is probably the best. I mean, the numbers.
All right.
Okay. Right, yeah, thank you, Jake. That's great. I think you need to run. I think we have Adam and Joshitza work out the details about the revised workflow and implement it. And I mean, that shouldn't be too complicated.
Perfect.
And then we move forward with those here. This is experimental details.
Okay, great. Thanks. Bye.
Thank you. Yeah. Bye.
Thank you, everyone.
Goodbye. Bye. So I would